Good morning, everyone. It's my pleasure to introduce Dr. Tharick Pascoal, who is the Neuroimaging Corps Oscar Scholar. He is an assistant professor of psychiatry who received his MD from the Federal University of Pelotas in Brazil and his PhD in neuroscience from the Montreal Neurological Institute at McGill University in Canada. He remained at McGill for his postdoctoral training in neurodegenerative diseases at the McGill Center for Studies in Aging and in neuroimaging at Douglas Hospital. Tharick's research program has focused on the development and validation of biomarkers capable of identifying pathologic processes in the human brain with the goal of developing therapeutic targets for neurodegenerative disease. Tharick has been awarded numerous awards. Most recently, he was awarded a 2020 Alzheimer's Association Clinician Scientist Fellowship. He also recently, since coming to Pittsburgh, has received his first 2 R01s, the first being the HEAD study, which is a quite large multi-site proposal co-led with Suzanne Baker at Berkeley to compare tau tracers head-to-head. And a second R01, a head-to-head comparison of high-performance plasma phospho-tau epitopes for the detection of Alzheimer's disease. Tharick's goal has been to submit a grant at every grant cycle since arriving in Pittsburgh, and he's been quite successful doing so and has several still under review and is clearly a rising star within my group. So it's my pleasure to hand it over to him.
Hi, first I would like to thank you for the introduction to Annie, and that she was instrumental in all of what she mentioned. And everyone here, I would like to thank everyone and say that it's an honor to be an Oscar Scholar and be considered for this and be talking to you here today. Well, she mentioned 2 projects, and I will present something a little bit different today. That is about the neuroinflammatory pathways of tau propagation in AD. And in this talk, we will kind of make a journey and what you have done in terms of microglia, astrocyte, and cytokines, and how these 3 processes are integrated or associated with tau pathology. I will start with microglia and say more the work that was done in Montreal. And then after, I'm going to migrate to astrocyte and cytokines in complements. That is the work that I have been done here. And I think it just is a sequence of work and makes sense to present together. Well, we will start with microglial activation. And here for microglial activation, I will ask you to follow me in a hypothesis-driven kind of thought. And this thought is start with some very well-known concepts. And the first concept is that microglial activation, and something that everyone knows, and this is not contestable, or at least the literature is very, very robust on that microglial activation is part of the immune response in the human brain. And that have been postulated as a key element associated with ID. I think this is pretty well established. Now we start to we'll start to fade how established it is. It will be at every time less established, and then we're gonna make our point. I think recently there are a lot of the literature that support by first for animal models that microglial activation is involved in the tau tangles propagation in AD. Somehow is involved. We know for human studies that the tau propagation occur more or less following some stereotypical stage in the brain called Braak stages. And this we know from post-mortem studies. Well, we know these 2 things. We know that there are 2 concepts here. One new concept that microglial activation is involved in the tau propagation in AD, and the very old concept that this tau propagation occurs in Braak stages. However, what no one established or no one showed before is whether there is a possibility of this microglial activation be involved in the way that tau progresses in the brain, the human brain, in Braak stages. And why this would be important? This is important because it will help to explain one of the most important unanswered questions in AD, that's how tau propagates and lead to dementia symptoms. Well, First of all, before talk about our results, it's important to mention that for do such a study that we're going to propose here, that see how microglia plays a role in our propagation, we need two things. We need a way, because when you talk about propagation, we talk about propagation from one region to the brain to another region to the brain. And then you need topographical information. You need to see where the disease is in a region of the brain, in the other region of the brain. And you need another thing. We need temporal information, because if we're talking about propagation, something propagates from the time 0 from the time 1. Then you need these two informations. What makes, in this case here, fluid biomarkers not helpful in this case, because they cannot give topographic information, and pathology not helpful, okay? Cannot give temporal information. Then this makes the necessity for us to use some kind of PET tracer. Although not perfect, the PET tracer that you have, as I just mentioned here, a brain injury can lead to an inflammatory process in the brain. This inflammatory process in the brain leads to activated microglia. This is very well consolidated. However, what I didn't say is that in the outer of the membrane of the mitochondria of the activated microglia, there is a protein called TSPO protein. That there is a tracer that can bind to TSPO protein with high affinity. Therefore, the uptake in this tracer is considered a proxy of TSPO protein, which in turn is a proxy of activated microglia. Then you have proxies here. It's just for you to understand that you are not measuring the real process, you are measuring proxies, and the results come without the limitations associated with this. Then, using this tracer, we decide to test whether microglial activation measured with this tracer is associated with tau propagation backstage for the first time. For doing that, we use a triad cohort in McGill. We measure this TSPO tracer called PBR28, an amyloid tracer at baseline in 130 individuals. And we also measure baseline and 1-year follow-up of MK6240. That's a tau tracer. Then you have all these components that you need to assess our hypothesis. Our population was across the aging and the AD spectrum. One thing that's very important to mention, and is a point that I want to make, that this study was different of all the others done with TSPO tracers, is that one thing that you need to know is that these TSPO tracers have a very important caveat. What's the caveat? That there is a technicality in these tracers. When I mean technicality, I mean that there is no repercussion on inflammation, no repercussion in AD. This is only a technicality. That have some people based on a gene that have this TSPO protein have different conformation. This makes the tracer bind to this protein with different affinity. But this doesn't mean that you have more or less inflammation. And this creates some phenotypes. Creates people naturally for the tracers that have a low affinity for the tracer, a medium affinity for the tracer, and a high affinity for the tracer. And this has nothing to do with inflammation. This is only the way that the tracer binds to this protein. And this is a big problem. And how everyone approached this so far? They normally take the groups that have a medium affinity and high affinity, they put together, because it's very difficult to get, they only have high affinity, and they solve this statistically. What you did here that, unlike everyone else, first to arrive in our 130 subjects, we start almost with 1,000 people, but we genotype 500 people. And then with exclusion, inclusion criteria, we end up having 130 people that have only high affinity binders. Then the study that they're going to present, the results that they're going to present, he's the biggest study ever done in a neurological disease with a microglia activation per se. And he's also the first study that you're going to take only the high affinity binders. Therefore, I know that there's a lot of preconceived concepts about microglial activation tracers, but I'd like to mention that it may be worth to see these results because this was the largest ever and was the one that have concern with the genotypic characteristic. Well, here is a little tough to explain, I will try. our first question was whether this microglia in the brain was kind of progressing or following a Braak stage-like pattern. There is a concept that comes from fMRI, comes from FDG. That's something maybe can be a stretch in some points, but this is the concept well validated and is not proposed by us, that the regions that are correlate with each other have some kind of, let's say, physical connection, and you can kind of infer a pathway that the pathology measured by these biomarkers are followed. This is an established concept. If someone thinks that's a stretch or not, it's another thing, but this is a concept that's out there. What you decide to do, take this concept from fMRI, from FDG, and put in the microglial activation trace. Then our question is, okay, if our hypothesis is correct, and microglial activation is progressing in Braak stages, we're going to divide the brain in 6 Braak regions, because it's an order, 1, 2, 3, 4, 5, 6. And the regions will be connected respecting this order. For example, we're going to have the BRAC1 region correlate to only with BRAC2 region. The BRAC2 region correlate to only BRAC3, BRAC3, BRAC4, BRAC4, and not with the other BRAC regions. This is going to suggest a kind of pathway that this is followed. And you test this hypothesis. And then you find that BRAC1 region was correlated only with BRAC2 region. BRAC2 region only with BRAC3, BRAC3 with BRAC4, BRAC4 with BRAC5, BRAC5 with BRAC6, and not with the other BRAC regions. Well, confirming our first hypothesis, well, seems that there is, and there is no reason for these regions be correlated like this. You know? There is no proximity, no reason for, at least we had the hypothesis, and the hypothesis was checked. Okay, this was the first suggestion that maybe this have a Braak-like pattern of progression for microglia as well. After this is, no one did this before, but this is not new because this is a hypothesis tested with a very large post-mortem literature, and we found the same thing for tau tracer. There was a Braak pattern of progression. And here, interestingly, further supporting what we hypothesize, here in this figure, in the y-axis, we have the components of this matrix, the components of the microglia matrix. The x-axis, we have the components of the tau matrix. And as you can see, the components of these two matrices was very highly correlated, suggesting that whether the connection of microglia activation was higher, the connection of tau was higher, suggesting or further supporting that there not only two seems like progressive Braak stage, but looks like there is a connection. When one is more highly connected, the other is more highly connected as well. But the real answer, or the most important finding that was here, a little bit more complex. I don't expect that you understand the whole details because I'm not going to have the time to explain the whole details. However, in general lines, you can see that we also found that the longitudinal tau propagation follow Braak stages. In our regression models, suggest that was the elements of the microglia activation network, rather than the tau, the baseline tau network, that determine the patterns of longitudinal tau propagation of tau. What this, like I said, general lines, rather than try to understand the details, but the general lines, this suggests that the most important component for the tau propagation was the microglia, how the microglia was disposed in the brain, rather than how the tau was disposed in the brain. Because what I didn't explain, that the main theory suggests that the tau propagates by spreading. Spreading by itself is spreading. We are suggesting, no, there are another element here. And maybe this is microglia. Well, but we are talking about that microglia is important for tau propagation. But then amyloid, how amyloid plays a role on this. And then what we did here, we can see here in the y-axis, we have the percentage of the brain affected by tau pathology. In the x-axis, we have the level of microglia activation in the brain. And in the barcode, in the colors, we have the level of amyloid PET pathology. More red, more amyloid PET pathology. And as you can see in this graph, are the individuals that have not only the highest microglia activation in the brain, but always the highest amyloid pathology in the brain that have the highest percentage of the brain affected by tau, suggesting that amyloid plays a very important role here, potentiating this process, accelerating the effect of microglia in tau propagation. Well, as you saw, results suggest that these three processes are important. Amyloid is important, microglia is important, and tau is the end point, and everyone knows that's important for the disease. But our question is, how does this play a role in the symptoms of the disease? Is this important for the symptoms? Have these three processes in the brain? And here, what you did here in this first figure, you have the codes here. White means no pathology, zero pathology. Here we take the individuals, every single individual, and we measure if these guys have all this pathology in the brain. And here is the percentage of individuals with, I'm going to describe the pathology, but the percentage of individuals with this pathology in these columns here. White means no pathology. This green means only amyloid, nothing else. Yellow means only tau. Orange means only microglia activation. This light green means tau and microglia activation. The blue means amyloid and microglia activation. Purple means amyloid and tau. And red means the three pathologies. These are the percent of individuals with the three pathologies. And as you can see, I should have done this before. And as you can see, the presence of these three pathologies was progressive increase from cognitive young to elderly to MCI to individuals with AD dementia. And when you do a logistic regression, we found that in this cohort, the presence of the three pathologies, amyloid, tau, and microbial activation, rather than only amyloid and tau, was the highest factor associated with cognitive impairment. Suggests that these three processes are interrelated. Amyloid, potentiating microbial activation lead to tau. And when these three processes converge in the brain, that the dementia symptoms accelerate. And this, we finalize the paper showing this model. In the right side here, what matters, suggesting that the activated microglia potentiate the tau spread or the neuronal tau spread from one neuron to another. And what I put here, this figure, one thing that's important, this figure that I'm going to open for the next studies, is this points here. That, you can see here cytokines, cellular receptors, complements, and you can see that this is generated by activated microglia, but can also be generated for reactive astrocyte. And this process ends up in CSF. And this process leaks from the CSF sometimes to the blood. And this opened the new line of work that you present now, that this is not mine, in fact, are from people that we work together, that are about astrocyte reactivity and cytokines and Alzheimer's disease pathophysiology. And this, just don't forget that these things we are not talking about microglia here, but we have worked on microglia, but just to be more focused on this. But these things are in fact processes that are end up in CSF and may leak to the plasma. Our brain process that we are talking here, not talk about here peripheral process. Then we will start with the work of Pamela. And Pamela saw in the literature that, as Nathan knows very well, that amyloid in plasma is highly associated with brain amyloidosis, on the left side. She also notes that plasma p-tau is associated with both, with brain amyloidosis and tau pathology. And there are a lot of papers now talk about GFAP. GFAP is a marker of astrocyte reactivity, showing that the GFAP is highly correlated with amyloid pathology. Then to do a very biomarker methodological related work, her question is, okay, a lot of people are saying that GFAP is highly correlated with amyloid pathology. But if I do a head-to-head study, how well GFAP is going to do in face of these other two pathologies that are theoretically measuring the real process? If you do a head-to-head, GFAP will be better, will be worse, will be the same. Then in the first head-to-head study with these markers, she found, I'm gonna just say in general lines, maybe she's gonna present her work better. In general lines, she found that while it's true that p-tau is the best marker for both, for amyloid and tau pathology in individuals cognitively impaired, GFAP did better than all the other markers we have here. I don't know if you have everything, but we have here p-tau 231, p-tau 181. We have, this is not complete, he has a more complete table. Now she has NFL, she has a beta 4240 using Simoa and using mass spec, both. And the results remain the same. She found that in cognitively impaired, p-tau 231 had best performance than the others. And in cognitively impaired individuals, GFAP did better for predict amyloid positivity and tau positivity. Then it's impressive. This marker is not really measure what amyloid and tau are measured, but the marker does better. Why I put this as a biomarker? For I remember of the difference here. There are two things that a biomarker can do. One, the biomarker can give a clinical information for you. There are amyloid in the brain or not. There are tau pathology in the brain or not. This is very different from, I'll talk about the pathophysiology. This is not the same. And this is not the same when she did this model. You can see that GFAP is very well correlated with amyloid PET, of amyloid pathology. Seems that there is a pathophysiological connection here. But when you correct for amyloid, the correlation between GFAP and tau disappear, suggesting that probably this occurred because this correlates with amyloid pathology. But as I say, this doesn't matter. GFAP is still a very good marker to identify tau pathology as well, but does not look like that GFAP is associated with the process tau pathology, because looks like this is more related to the association with amyloid pathology that leads to association with tau pathology. Then going a little deeper, GFAP, okay, we identified that GFAP is associated with the process. It looks like it's associated with AD, with amyloid pathology. And then the question that Douglas here want to answer, and he has more complete data as well, this is a less complete thing, he's dividing cognitive domains. He wants to assess if this plays a role in cognition. He's dividing several other cognitive domains and neuropsychological symptoms. And here's just to exemplify that GFAP seems more correlated with memory than with executive function and language. This is important information about what this means. When you correlate with different cognitive domains, we in fact are correlated with different brain regions, because memory is more related to the temporal lobe, more medial temporal lobe, executive function more in the frontal lobe, language more of the temporal lobe in the left, more superior. This suggests that there is some localization of this GFAP, there are some localization of astrocyte reactivity. This suggests that maybe a study or a PET scan able to detect this and give topographic information can be very valuable, because maybe there is information out there. There are markers that the global value and the regional values are more or less the same, and you cannot predict. You're only going to know when you have PET scans to see that. For example, there are a lot of people that say that amyloid PETs are more global and doesn't matter if I have regional. Tau is very regional. Microbial, we show that's regional. Astrocyte activity, we still are not sure. And these results kind of suggest that you're going to have results that they're going to propose that they have a regional information. And then here's João. If the question is, I'm going to need to do faster because my time is probably gone. The question that João did is, okay, we know that GFAP is associated with amyloid and not associated with tau physiologically. But then this means that astrocyte activity is not associative tau. No, this doesn't mean that, because I have another markers of astrocyte activity such as YKL40 that seems more associative tau. He found that while is correct, that GFAP is the marker more associative with tau, he's comparing here with many cytokines. YKL seems the marker that's more associated with amyloid. Then doesn't mean that the astrocyte activity is not associated with amyloid, but mean that that target is not associated with amyloid. And here in this graph that I'm not going to have the time to explain, but here in this graph, he can show the size of this means the fold change between amyloid positive and amyloid negative. He can show that where GFAP show the highest fold change between amyloid and tau positive is not as very much associated with any other process in the brain. I'm going to explain after how the process is associated, suggesting that this is a kind of a process apart in terms of the inflammatory pathways, suggests that can be a signature of amyloid deposition. And here he found the same to tau. Sorry, I'm going to need to be fast that my time is running. But the tau, he found the same thing for YKL40, suggests that this may be a signature for tau. However, João said that the cytokines here show many cytokines, GFAP and the other proteins. And he said that, okay, these are much better markers for amyloid and tau pathology. However, Bruna tried to prove ring wrong. And we measure 96 inflammatory related proteins in the brain, that cytokines. And we did a bit different approach here. First, like João said, is correct. YKL is the best marker, and the other cytokines are not very well related to tau pathology. He's correct. However, she tried to do something differently. She takes these 96 inflammatory related proteins. I'm going to explain in the next slide how she did that. And she divides this 96 protein in 7 clusters based on physical proximity. You're going to see after how she did that. With the 7 clusters, let me explain first how these clusters work. She took this 96 cytokines and she did a model. How she feed this model? She feed this model. We know that there are a lot of inflammatory processes in the brain, and this process, there are a lot of papers showing how this process works and how far are from the proteins from each other. How she clustered this data. What means the connection between this region? This connection between this region means the average of the proximity between these proteins in all the inflammatory processes we know in the brain. Then what the model does is clustering based on physical proximity. Here are the processes. I cannot read this because the people, my time is going, but here are the different processes we can discuss after. And here is, and these lines here mean the average of proximity in this process. And then you cluster these proteins like this. Okay, I can explain only this now. Once she did that, she clustered this, she divided, she take the 7 clusters and average the value of the proteins in each one of the clusters. Before, the proteins alone could not differentiate individuals' plasma, p-tau positive and negative, when you divide in positive and negative. But when she did these clusters, you can see that 2, 3, 4, here, 4 of these clusters, of the 7 clusters, were able to significantly differentiate individuals tau positive and tau negative, measured with plasma p-tau 181. While famous markers like GFAP, YKL alone, and no other cytokine was able to do that. Suggesting that this new approach that she's proposing, that in fact analyze these things more as average of pathways, because everyone knows the term inflammatory cascade. As a pathway of as average of the cascade can be more robust than the simplest average of a single target, because you know the noise that these things have. When you average them out, the noise decreases, the data gets lower standard deviation, the correlations and the difference appear. Well, these findings as a role suggests that neuroinflammatory pathways are key players in the pathophysiological progression of AD. And I'd like to thank everyone here in Pittsburgh and in McGill for the data. Thank you.
Tharick, what you're presenting are correlated observations in the individual, where you also have group differences.
Yeah.
And so how do you try to, if you went back to one of your correlation plots, how do you try and think through the difference between a correlation that's driven by the group differences where you get like the dumbbell tilts versus a real, a correlation maybe more indicative of a biologic process?
I think this is a very good question, and is a very famous thing about the correlation, difference between correlation and causation. And I don't think with this type, we can try many ways, but I don't think you can address this in a way that will be very compelling. I think the only way is doing intervention and doing mechanistics thing. What Bruna hope, this is not my expertise, but it's her expertise. What Bruna hope is try to identify the pathways here for really test, as you were saying, if you have some causation with knockout, with something that you're going to give a little bit more proof of causation after, she's just bringing the hypothesis, and you are completely right. About the difference between the correlation and the causation, and how this is related with a pathway or is related with the main framework of the disease. But we can try to address this with grouping, with some techniques, but I think address this in a compelling way only with mechanistic experiments that will come after. I agree with you. Thank you for the question.
I'll ask a question. That was very interesting and exciting work, looking at how neuroinflammation plays this key role. And, it really is begging this question, where can we go? How can we intervene? And one of the arguments I had with Oscar a little bit is, I always wonder if we're trying to decrease inflammation, we know these lifestyle and behavioral factors do things. And it's actually our biggest recommendation now is, good sleep, diet, we see these effects. And then we have these complicated treatments, pulling out amyloid, plasmapheresis. Do we think maybe, I wonder if you looked at the effect size, if you did a really good behavioral intervention, if maybe the effect size would be as good, if not better, and the risks would be much lower?
I agree. I agree 100% with you. But let's imagine here a line of a research, how the things plays out when you do something like this. I don't think we still first, for example, like was amyloid. There is lots of research that start here with the PIB that's showing very compelling results, and no one have doubt that there is association between amyloid and AD. No one have doubt that there is association. Association, no one can contest. Causation, like you said, no, I don't know. But association, no one can contest. I don't think we are still in the stage we know or we know very well in a compelling way what's the factors that play a role in the neuroinflammation. And here comes is another study that you are planning here and you're proposing. We are trying to do a multi-site, multi-country study where, exactly like you said, we will try to address all the factors that play a role and the effect size that they play a role in neuroinflammation.
What I would argue is you need a head-to-head comparison. And you can't say, let's give everyone the best behavioral intervention. And then let's look at how much the extra expensive and pharmacologic and potentially dangerous interventions have an improvement over it. I think it's important.
No, I understand the comparison head to head, but do you think you already have what I mean is the step before this? Do you think you already established that there is a very strong link between brain inflammation and this behavior intervention? No, it's for this reason that. No, I agree with you. I agree with you. I agree with you. For this, what I think is that maybe we need studies, large studies, to correlate these things as we are planning to.
Don't just take the natural. You have to give as good a shot to the behavior as you do to the.
But we think we are quite there already. I don't know. Maybe I don't know the literature. We think we are quite there. We know very strongly that there is a link, not only with inflammation. It needs to be a measured inflammation. It needs to be these markers, because if not, you don't have a basis. Everything will be a hypothesis. Like I said, maybe you're completely right, but maybe the markers that you have available, the technology today, you cannot address them. And then you cannot have nothing for clinical trial surrogate. you know, do you think it's established already that, for example, sleep? We are, like I said, we're planning a study that we're going to address on other things like pollution, a lot of stuff. I didn't have think sleep. I think it's a great idea.
No, I mean, I think it's.
But do you think we have this established already that?
I think it's established enough that we all tell our patients that.
But that, no, I understand. But do you think the markers that you have can detect that?
As much as they can with perhaps plasmapheresis or adjuvant.
Okay, then it's a. No, I'm not saying anything.
Things that are right for information, my understanding is at this point, it's sort of a biphasic relationship to pathogenesis. Meaning that there's protective and
Yeah.
like exercise.
Yeah, but your point, I think, it converges with what I said for him. We don't have even established where how this biphase work. I don't think you are, like I said, you can contest at the helm, you can contest anything in this point, but you cannot contest that you understand the association.
I understand, but
You have a robust biomarkers, you have robust results. I think inflammation is not there in the same stage. You know, I think you need to bring inflammation for this stage. Okay, now it's in the same stage. Now we have more studies after you do the.
Well, regardless of what the biomarkers we're looking at, in terms of its effect on AD, the behavioral and lifestyle interventions seem to have perhaps a similar effect size.
You are talking about cognition straight away, without any biomarker in the middle. Any biomarker in the middle.
And I think we ignore that and just focus on where the.
Okay, then I agree with you. If you see, I don't have the. No, I don't have the same knowledge than you in this thing. I don't know the literature very well. But if you are saying that, yes, it would be very easy to do if you take the biomarkers from the middle and see a cognition intervention with Aduhelm, head to head is very easy to do now.
No, I get, yeah, the point, yeah, I think we have maybe not.
No, I understand. I agree with you. I agree with your point in general, but with the inflammation in the middle, kind of not, but with the general, I agree.
I just have a bit along the line of like biomarkers, especially like clinical diagnosis for biomarkers. Is GFAP the best biomarker to have? I think of this in kind of its context of like systemic lupus erythematosus, in which you have ESR and CRP, which are sensitive but not specific.
Mm-hmm.
So GFAP is just telling you that there is inflammation, but it's not telling you what the inflammation is from, or is it that specific to AD?
First thing, I don't think is specific of AD, but just to clarify that our models are under the curve, that they take the sensitivity and specificity of everything, you know. And I know that our cohort is not the best. We have FTDs, but it's not some very broad cohort. Maybe I would be nice to see in the real world how this will behave, but our data consider both sensitivity and specificity. And of course, probably that's not specific for AD. However, what you measure there, like I said, as a biomarker, is doing better than plasma p-tau and plasma A-beta. This is a just simple comparison, it's a fact. I agree with you with your consideration, but was a fact. Unexpected, I don't know if this is the results that you want, but these are the results that appear.
So everyone has a few minutes on the Q&A. If you could turn your attention to the Q&A.
Q&A here. The first question from Bill Plank. It's possible to see a similar pattern of topographic association of microbial activation if you do a voxelized analysis with seeds in each BRAC region, as do with a simple BRAC regional analysis. This is a very good question. I didn't do that. I don't know the answer, but I would say that this is an interesting question because, in fact, there are a person that is doing a kind of opinion paper, and this person asked me this data to do a seed network analysis to put in the paper. I think we're going to discover soon. I'm going to send the data to her, and this is a very good question, but I still don't know the answer. I would say that probably yes. Seem very robust, and I would say that probably yes. Julia, Tharick, I see most. Julia asked about that she saw that, I think in the same line that the question that was made here, that I see most astrocyte activation in AD patients that occur together with TDP pathology. If there are some histopathological studies with GFAP, I didn't see yet. There is a way to do a very easy one here because GFAP is now available in ADNI some weeks ago, and ADNI has some pathology will be few people, but I think you can have, we could find result with p-tau. This is a very good point, because if you find, then I know that you have at least 17 patients that are going to have pathologies FAP, because they are analyzing the same sample. I know the sample. I think this is a very good question. It can be tested very easily, because the data is there, is available. It's just to do the correlation. With p-tau, we find a very good correlation. Then looks like one is better than another, but I don't know if the pathology is going to confirm this. I will try that, Julia. Thank you for the suggestion. I'm going to send you the result.
Thank you, Tharick.
Thank you.